Okay, so students, I think we can start the class. So what we have discussed in the last class was we were talking about in the first part of the lesson. What we have discussed, we have talked about the sexual part of a plant, that is the flower. Whenever we talk about the flower, what we remember that it is made up of how many holes? 4 holes. What is a hole? A hole is nothing but a collection. Right? We have 4 holes, and the 4 holes are calyx, corolla, androecium, and gynoecium. From sexual point of view or reproduction point of view, which 2 holes are important? Obviously, the essential holes are important as compared to the accessory holes, right? When we talk about accessory holes, which are the holes we are talking about is calyx and corolla, and if you're talking about the if you're talking about the essential, we are talking about androecium and gynoecium. Talking about androecium is supposed to be the male part and a female part, obviously. The male part of the plant, and gynoecium, the female part. Androecium is a whole, it's a collection, or individual members of androecium is called as what? It's called as stamens, right? And individual member of gynoecium is called as carpels, or it is called as pistil, right? Since we have to talk about reproduction, so obviously we are concerned with the stamen and the carpel. So in the last class, what we have discussed is, we have talked about the stamen. Whenever we talk about stamen, a stamen consists of how many parts? 2 parts, that is the anther and the filament. Some points about the anther that we have discussed was, Kiki, anther Kiki mondase. Bilobed, right? We have talked about words like monothecous, dithecous, right? We have also used words like bisporangiate, tetrasporangiate condition, etc., etc. So what we remember is whenever we talk about, androecium boli kole, we remember stamen. And when we talk about stamen, we have anther and filament. And when we talk about stamen, we have discussed 2 types. Last class or common revision. Dithecous, monothecous or diagram, a rough diagram. Dithecous or ki akisile enekwa no? Right? So this is the anther part, this is the anther part. Inside the anther, what you will see is pollen sac or microsporangium. Yate keita ase, duta, hekane condition ki? Bisporangiate, yate ki asile? Tetrasporangiate. Which condition is most commonly seen in angiosperms? However, we have talked about certain exceptions also. We have spoken about Malvaceae, right? Okay. Next is, after this, what we have studied is, we have talked about yes, do you remember? Right? So when we talk about this one, when we go for a TS of this bilobe or dithecous anther, we get a curved structure. Right? And when we talk about the TS, what we see? Tissues. The outermost layer of tissue is called as epidermis, right? Epidermis. Below the epidermis, the layers of cells are called as archisporial layer. Okay. And next, what we have said is, in the archisporial layer, one cell gets, it becomes big, and that cell would be called as archisporial cell, right? So archisporial layer or ki hoisile. So if you talk about here, one of the cell will become big. Similarly here, here, and here. And this would be called as ki archisporial cell, right? So notun ke jun ahisa okoman idea paisa what we are discussing, right? So you need to remember these words, bisporangiate, tetrasporangiate, etc. Or jihetu we have to talk about reproduction, by sexual reproduction. What we are in search of is something called as gametes. Gametes laibo, male gamete, female gamete laibo. So male gamete kot thakibo? pollen grain ভিতৰত থাকিব pollen grain to ক'ত থাকিব pollen sac ৰ ভিতৰত থাকিব আৰু pollen sac to ক'ত থাকিব anther ৰ ভিতৰত থাকিব right so that's the reason why we are talking about the TS so that we can study the pollen sac pollen grains anther to ভিতৰত কেনেকৈ form কৰিলে right okay okay then what we said if suppose we are talking about sorry etu color distinct nohoy right so what we said if this is the epidermal layer we have talked about a cell that is called as archisporial cell what will happen in the next stage this cell will undergo division. What type of division? We have used two words, no? Either it would be anticlinal or it would be periclinal. Archisporial cell will undergo division and form two cells. Ki asile? Archisporial cell will undergo what? Periclinal division will form two cells. The one towards the center is called as inner sporogenous cell আৰু বাহিৰলৈ থকা সেলটো কি কৈছিলে outer primary word to yet inner primary okay outer primary what was the name parietal cell I think we remember this part also right so we have talked about this archisporial cell dividing into two cells periclinally we have two cells one is called as the outer parietal cell and the other is called as the sporogenous cell what we were discussing was the development of the parietal cell in the last class right so jihetu a2 cell our cell or ki tendency thakibo obviously cell has a tendency to divide no so a2 cell divide kori pele ki form korise duta cell form korise similarly these cells will also have tendency to divide so the next thing that we have studied was the division of the parietal cell right so primary parietal cell will undergo division কেনেকুৱা ধৰণৰ division হ'ব এবাৰ both anticlinal and periclinal both type and ultimately they will be forming right so eta cell e kuntu cell bahirot juntu cell asil tak ami ki boli koise parietal cell divide and redivide kori kori ultimately ki form koribo layers of cell and that would be called as the wall mane ki hor right pollen sac wall kon form koribo right what we were discussing was about this one when we talk about this layers no yate ultimately ki form koribo yate next diagram tut parietal cell ki koribo divide koribo no divide kori pelai kiman layers form koribo 2 to 6 something like this they will be forming layers so outer layer we have discussed in the last class was called as ki epidermis single layered and multiple layered obviously single layered next layer that we have discussed endothecium single layered and multiple layered single layered one very important point about endothecium that we have discussed the presence of dash alpha cellulosic fibrous bands right right next is 2 to 6 layered hobo lagibo no eta lage ami keita layer porilu duta layer he porilu tar pisot to aru layers thakibo right so the next one that we are going to study you have to start writing from that I think we have given this as number 1 this as number 2 right so the number 3 what you'll be writing today is middle layers Is the point understood, right? Ami kihor upor information likhisilu, ami epidermis information likhisu, right? Endothelium information likhisu. Below the endothelium, what you'll see is the third layer that is called as middle layer. So we'll be writing points about the middle layer. So heading koriba middle layer. So parietal cell divide kori pelai ki form korile, layers of cells form korile. The outer layer was called as epidermis, inner to epidermis, endothelium. Inner to endothelium, we see multiple layered cells. Everything together was called as middle layers. What we have to know about middle layer is the cells over here are loosely arranged, and whenever we talk about loosely arranged cells, we understand that intercellular spaces are present. Intercellular space buji pao, duta cellular majo junto space, no? Not necessarily each and every cell will have intercellular spaces, right? We have cells that are tightly arranged, we have cells that are loosely arranged. So in this section, what we have studied is the cells are loosely arranged, first point. Second is, what type of a cell are we talking about? We are talking about parenchymal cell. As far as all these cells are concerned, parenchymal cells or sclerenchymal cells, I think this is something that you have studied during anatomy in your first year class. Anatomy purisa nai. Anatomy purisa, obviously it's a part of your course, right? Plant anatomy, obviously you have started with tissues. The word tissue, when you talk about plant tissues, no? Broadly, kiman type of? You can divide the tissues broadly into how many groups? 2 groups. One was meristematic, the other was permanent, right? When you talk about meristematic, we know the characteristic, the special characteristic, it will keep on dividing and redividing, right? If you're talking about permanent tissues, why they are called as permanent? Because they will stop the process of division, first point. Permanent tissues are classification, how many types? 3 types this time, right? Ki asile? Parenchyma, collenchyma, sclerenchyma, right? And out of these 3 tissues, universal tissue boli ki kakom, something that's present almost everywhere, that is called as universal parenchyma, right? So most of the parts of the plant body ki thakibo parenchyma tissues thakibo, right? Next, middle layer, we have used another word, ephemeral. What is ephemeral? Short-lived. Tamane out of all these cells, what will happen as the anther matures? Hekini cells mori goi thakibo. Right? Next, inner to the middle layer, we'll have another layer that is called as tapetum. Have you heard about this word before? Tapetum. Whenever you talk about tapetum, what is the function of tapetum? Because tapetum boli kole ami function to monot rakho, right? So tapetum is also important function ki nourishment, right? So we will talking about groups of cells that is called as tapetum. Tapetum or function, obviously nourishment, but apart from nourishment, they will have another function that is called as secretory function. They will be forming certain things. Okol je nourishe koribo, not necessary. Kisuman bostu form koribo. Ekane function boli kole, duta type of functions will be studying, but what is the most important function of tapetum? It's obviously to provide nourishment. Kak nourish koribo? Ami ki pori aso? Pollen sac or wall khini. right? Inside the pollen sac, what you'll have? The pollen grains, right? So you have certain cells that will ultimately form the pollen grains. To form the pollen grains, they will be requiring food. Food kune dibo, tape, right? So we'll write the points about tapetum, then we'll draw the diagram. So rest of the layers, as far as epidermis, endothecium, middle layers are concerned, I think we can understand from the figure, right? Inner to endothecium, the single layer of cells is called as tapetum, denoted by the blue color. Inner to the tapetum, we have, remember that cell called as sporogenous cell. We have talked about two cells, right? First, we have started a discussion from a cell called as archisporial cell, right? We have talked about this. Suppose this is the archisporial cell. The next diagram was archisporial cell divided into this one called as the sporogenous cell, this one called as the parietal cell, right? After that, what would be the diagram like? If you are talking about this parietal cell, it will divide and divide and divide. Ultimately, they will be forming many layers. What will happen to this sporogenous cell? This will divide and re-divide again, and ultimately they will form tissues. Is the point understood? The sporogenous tissues kune form korile? Amar cell keta asile duta, parietal cell aru sporogenous cell, right? So parietal cell divide and re-divide kori pele layers form korile. Sporogenous cell tio to divide koribo. Sporogenous cell initially ki asile single cell asile. He divide kori pele multiple cells, or ultimately if you have a collection of cells, we'll be using a word key. Tissues. Or no, hekane pothomote ki likhisilu sporogenous cell. Etia ki likhisu sporogenous tissues. Is the point understood? So who formed the sporogenous tissues? Obviously the sporogenous cell, right? Our sporogenous cell to kot asile, inner side of asile na outer side of asile. Inner side of asile. So obviously it will be covered by that sac wall, right? Talking about the tapetum, innermost layer, I think from the diagram we understood, right? Sporogenous tissues of, one is the last layer of the wall. It is tapetum, right? Next is surrounds the sporogenous tissue. Who formed the sporogenous tissue? We understand. Talking about some characteristics of this cells, tapetum ki hoy? A layer of cells. A layer of cells, obviously we understand it's not made up of a single cell. So what we are writing here is characteristic of those cells. Tapetal cells bilakor ki thakibo? Large nuclei thakibo. Aru ki thakibo? Thick cytoplasm thakibo. So ei kita point about question ahe. Etu upra question ahe. Normally they are multi-nucleated. Etu point upra question ahe. Tapetal cells are polyploid. Etu point upra question ahe. They have a huge amount of genetic material, and obviously most important function is nourishment, right? From these points, whenever we talk about a cell having a large nuclear and thick cytoplasm, it is a characteristic of dash cell. When we talk about cells or tissues, two types, meristematic or permanent. Large nucleus, thick cytoplasm is a characteristic of permanent meristematic. Current anatomy, you have talked about certain characteristics of permanent tissues, certain characteristics of meristematic tissues also, right? So whenever we talk about a cell or a group of tissues with large nuclei and thick cytoplasm, dangor ke nucleus to kyo lage, basic cytoplasm kyo lage, division or karne. Because if a cell has to divide, it has to divide its contents into two equal parts. So obviously it should have huge content, right? So these are the characteristics of meristematic tissues. Right? Next is they will be multinucleated. We know again cell theory is something maybe you all remember. Cell theory ki koisile je each and every cell will have a single nucleus, but we had certain exceptions. If you remember, kisuman cells or to nucleus na thakei. Kisuman cells or etatke besi nucleus thake, binucleate condition o thake, multinucleate condition o thake. So tapetal cells are multinucleated. Next is a word called as polyploid. Have you heard about words like haploid, diploid? Right? Haploid cell, diploid cell. Haploid, diploid and polyploid. What do you understand? Eta cell ok haploid buli ketia kom? When it will have one set of chromosomes, right? And a cell would be called as diploid if it has two sets of chromosomes, one from the father, the other from the mother. Duita jedi ekelege thaki pele eta pair form kore. Protective cell or then it will be called as a diploid cell. And from the word itself, polyploid mane many sets of chromosomes, right? Sometimes what happens, a single cell might have many sets of chromosomes together. If suppose a single cell has many sets of chromosomes, whenever we are using the word chromosome, we are talking about the genetic material, yes or no, right? So whenever we talk about genetic material, we are talking about key DNA and RNA. So jedi genetic material besi thake, jedi chromosomes besi thake, tetihale DNA besi ke thakibo na thake. Is the point clear? So, question ahibo pare je increase in DNA content is a characteristic of tapetal cells. Polyploidy is a condition seen in tapetum cells. So, these are the words that you will focus on. As far as the function is concerned, as I said, most important function is nourishment, kintu okol nourishment e nohoy. So, the next part we will be writing it down. Ekini hol? So, tapetum will continue. So, talking about types of tapetum, kiman types? 2 types. Naam gita swason, one is called as secretory, the other is called as amoeboid or plasmodial, right? Whenever we talk about secretory, what do we understand? That maybe these cells will be forming something. Kiba kibi secret kore, hekane tak ki boli koiso secretory. So we understood that all the cells of the tapetum ki hoy? At a layer of cells, right? All the cells of the tapetum are not responsible for nourishment. Some of the cells of the tapetum will be secretory in nature. So ki form kore, we'll be writing it here. The next part, we have a word called as amoeboid or plasmodial or invasive tapetum. We will see later that this is the type of cells that would be providing nourishment to the sporogenous cell, right? Kisamane nourishment koribo, kisamane kiba kibi bostu form koribo. As far as this word is concerned, amoeboid, obviously amoeba-like. Do you remember this word, plasmodial? Plasmodial word to quote porisa question. Remember these two words called as plasmodium and pseudoplasmodium. Plasmodium, pseudoplasmodium, ki asile? Aggregations asile. Kar logot associated? Slime moles. Slime moles porisile no? Protistot. So slime mole ki asile? A fungi-like protist is called as, one example is a slime mole. Slime mole bilake keneke thake? They'll stay in a team, right? That arrangement, depending on the type of arrangement or depending on how they are staying, either that formation would be called as plasmodial formation or the formation would be called as pseudoplasmodial formation. So here we are using this word plasmodial and amoeboid. Plasmodial formation to kenuga asile, if you remember from slime moles, no? Slime moles bilak, right? Sometimes what happens, each and every organism will fuse and ultimately they will be forming a multinucleate structure, yes or no? Right? So individual eta asile tahate fuse kori pela multinucleate structure form korile, a aggregation to kami ki koisil? Plasmodium, right? Plasmodium formation. So we are using the word plasmodium. What does it talk about tapetal cells? It says that some of the tapetal cells will fuse together and ultimately it will form a multinucleated cell that would be providing nourishment to the growing sporogenous tissues. Right? So what we have discussed up till here, when we talk about tapetum, it's a layer of cells. Some of the cells of the layer provides nourishment. Fuse koribo, gutekini contents kak di dibo, belegok di dibo. Beleg mane sporogenous cellok di dibo. Aru kisumane ki koribo, kiba kibi bostu form koribo. And what we are talking about is, heard about a word called as sporopollenin. Sporopollen nahi khuna na? Okay, we'll be writing it down. Chalo. First part to likha. Yeah, SPOROPOLLENIN. UBH bodies, UBISCH. UBISCH. এইটো part বুজিছো right amoeboid tapetum but these cells তাহাঁতৰ কাম কি হ'ব nourishment কাক দিব microspores আৰু এটা নাম কি আছিলে pollen grains কোনে ফৰ্ম কৰিব sporogenous tissues বিলাকে right so now we understand the word microspores now as it is in the first class we have spoken about the words called as microsporophyll megasporophyll, microsporangium, megasporangium, microspores are megaspores, right? So whenever we are talking about microspore, we are talking about the pollen grain. If you are talking about microsporangium, it refers to what? The pollen sac. If you are talking about microsporophyll, we are talking about a leaf-like structure that has the microsporangium. What we are talking about is nothing but a stamen. Stamen ki asile? Modified leaf, right? Each and every part of the flower ki hoy? Nothing but modified leaf. Right? So, now we'll be writing certain points about this. Points puli likhiba. Okay, write it down. Oh, এটা word ভুল হৈছে. It should be granular, grain-like. লিখা হল? Okay. So, 18th point, or maybe you have come across a word this called exine. So, what is exine? All of pisar ako details likhim. What is exine? Whenever we talk about a pollen grain, the pollen grain has two walls. The outer wall is called as exine and the inner wall is called as intine. Right? So, we are talking about the outer wall of the pollen grain. UBH bodies ki hoy, so if you are talking about this as a pollen grain, no? Pollen grain or ki thake, dukhon wall thake. Enekwa no hoy, major pore we will discuss that later. Yate, kisuman, enekwa grain type substance thake, tahatok ami ki buli kom? UBH bodies buli kom, right? So, pollen grain or exine or kisuman grain-like substance thake, that is called as UBH body. What is the function of that? Thickening of the exine, aru thakar karone ki hoise je exine khon besi thick ho goise, and plus ornamentation. So, UBH bodies are kam ki, it thickens the exine plus beautification of the exine. Ornamentation, what to use korile, we talk about from the beauty sense, right? Jetia eta bostu thakibo, so we talk about it as its ornaments. So, UBH bodies are basi eku nalage. Iar pra jodi question ahe, hudibo je who is responsible for the secretion of UBH bodies? Tapetal cells are responsible for the formation, right? It is a characteristic, or it is a grain-like substance that is found in, it is a grain-like substance that is found in the exine of the pollen grain, right? And what is exine? The wall of the pollen grain. I think it's understood. Next, we are talking about another substance that is considered as most resistant. Resistant mane? Resistance against what? Right? Maybe resistance against diseases, resistant against germs, right? So, pollen grain too generally ki consider kore. Do we understand a word called as viability? viable pollen grain, viable seed, viable মানে কি যেটালৈকে germination part থাকে তাক আমি কি বুলি কওঁ viable, right? এটা pollen grain viability বহুত বেছি, right? For years together it can survive. If something can survive for years together, it has to be very resistant. Resistance against what? Resistance against many factors together, maybe because of the environment. বিরাট গরম হলেও তার problem নাই, বিরাট ঠান্ডা হলেও problem নাই, right? Germs-ৰ কাৰণে বা insects-ৰ কাৰণেও problem নাই, right? Who makes it resistant is a chemical compound that is called as sporopollenin. That is supposed to be the most resistant substance in the bio world, and that is the reason why pollen grains bilakor viability period বহুত বেছি. It's not like this that each and every pollen grain of viability period bahut besi. Later we will study that depending on plant to plant viability period varies, right? But in general, kisman pollen grains to enuka kisman ami examples porim. Maybe you remember something from the living world also. Living world or porhisa tumai. Kisman pollen grains enuka say that thousands of years it has survived. Right? It is viable even after 1,000 years. There are certain examples which remain viable even after 2,000 years. So 1,000, 2,000 years survive koribo lagile, ba ako germinate koribo lagile, obviously it has to be resistant. And who makes it resistant? Sporopollenin. It is something, it's a chemical compound secreted by whom? Tapetum. Right? Next, it forms the exine of the pollen grain. Obviously, gute exine khon kihere bonua? Sporopollenin. Aru hei karone pollen grain or bhitoror portion to ki hoi thake? Protected hoi thake cells bilak. Right? Aru sporopollenin uporot ki thakibo? UBH bodies grains or nisnake thakibo. Right? Next, the third one is something called as pollen kit. Before discussing this pollen kit, I think you all have seen the hibiscus flower. দেখিছা no? Hibiscus or junto anther part ase no? If you touch that, you see a yellowish powder that sticks to your hand, and this is nothing but pollen kit, right? Something that's sticky in nature, something that is yellowish in color, right? It is rich in all this, and that's the reason why insects bilak ahibo pollination or karone, right? It is found in certain flowers like hibiscus, we understood, right? It is present in insect pollinated plants. Out of all the points, this point is very important. Presence of pollen kit is a characteristic of, I think last class we discussed we see different types of pollination method in different types of plants, no? Kisuman plants or ki hoy, the pollination takes place by means of water. Kisuman or ki hoy, by means of air, kisuman or by means of insect. So we have biotic conditions, we have abiotic conditions that helps in pollination, right? Adult plant talk, okol insecte pollinate koribo. Adult plant khali water ote pollination hobo, depends on what? Depends on the characteristics that is present in the plant, right? So if I think in last class I have spoken about this, jedi air pollinated hoy tetale obviously flowers bilak ki hobo libo, light hobo libo, right? Thik teneke water pollinated jedi hoy pisot ami porhim tando ribbon like petiole thakibo, stigma thakibo, etc. etc. So in this case, if you have a sticky substance, hetu ki hobo? Anthero usort ki thakibo? Sticky pollen grains thakibo. Hey, sticky pollen grains bilak ki hobo? Insects gat lagi goi pelai, they will assist in pollination, right? So I think we understood what is a pollen kit. First, is it present in all plants? No. Is it present in all flowers? No. It is present in only insect pollinated plants, right? And one example of them is hibiscus. Jedi question ahe, hibiscus or a question ahise. Or নহলে the last line, right? Okay. We were discussing certain points. Point number 3 asile no? Ekini first note to likhi lom, then we'll explain one by one. Okay, so what we are writing here is অলপ আগতে এটা পইণ্ট লিখিছিলোঁ ন, one characteristic of tapetum. First polyploid লিখিছিলোঁ, second DNA বেছিকৈ থাকিব নে কমকৈ থাকিব, বেছিকৈ থাকিব, and what is the reason behind, right? Not very important from examination point of view, এতিয়ালৈকে ইমান questions অহা নাই, কিন্তু আহিবও পাৰে, right? So we'll just do it. প্ৰথমে লিখি লোৱা. So what we are writing is what leads to the increase in the amount of DNA in the tapetal cells. Tapetal cells বুলি ক'লে normal cells নিচিনাই হ'ব লাগিছিলে কিন্তু যেহেতু polyploid তো obviously তাহাঁতে DNA material বেছিকে থাকিব. And what could be the reason? Different plants undergo different methods to develop polyploidy conditions. বা কিবা কিবি কি হয় normal type of cell division নহয়. কিবা এটা অকমান unique type of cell division হ'ব that results in certain characteristics that leads to কি increase in the DNA content, right? Specifically, বহুত reasons আছে. We'll be talking about just 3 of them. তিনটা reasonৰ কাৰণে maybe যে tapetal cells or কি হয় DNA content বেছি হয়. The first one that we are talking about is endomitosis. No doubt we have written a lot. What you need to remember is কি লিখিলো এটো? Just a second. Okay, out of all the things that we have written, just read the one that is written within the bracket. Ki buzi paise swa. So what we are saying is, tapetum cells or DNA kiho karne besi hoise, maybe because of a process called as endomitosis. And what is endomitosis? Ekhini bad dia, a last line to swa. This line talks about a different method of cell division than the one that we have studied, yes or no? আমি যোনটো cell division method পঢ়িছো, cell cycle and cell division পঢ়ি আহিছা, cell cycle and cell division ৰ যোনকিটা method পঢ়িছা, তাতকে অকমান বেলেগ হয় নহয়? কি বেলেগ? Spindle formation form নকৰে আৰু nuclear membrane খন কি নহয়? ভাঙি না যায়. Right? Whenever we talk about cell division, either we are talking about mitosis or we are talking about meiosis, তাত কি হয়? প্ৰথমে nucleus divide হয়, তাৰপিছত cytoplasm divide হয়. Nucleus যেতিয়া divide হয়, এটা stepত divide হয়, we have 4 phases. 4 phases, what were the 4? Prophase, metaphase, anaphase, and telophase. What was the specific point about prophase or the most important characteristic that you observe in prophase? Nuclear membrane degenerates. Prophase বুলি ক'লে প্ৰথম লাইন আমি হেইটোৱে পঢ়ো ন যে nuclear membrane খন ভাঙি যাব আৰু তাৰ পিছতহে তাৰ পৰা ইভেন্টস বিলাক আৰম্ভ হ'ব, right? So this is a process where nuclear membrane খন ভাঙিয়ে না যায়. So all the events occur within the nuclear membrane itself. So অকমান to normal টোতকে অকমান বেলেগ. So maybe that this is the condition that leads to increased amount of DNA in the কি tapetal cells. যদি nuclear membrane খন ভাঙি না যায় তেতিয়াহলে কি হ'ব? Chromosome split নকৰিব. যদি chromosome split আর spindle fiber ও form কৰে নাই. যদি spindle fiber, what was the function of a spindle fiber? Poles. Just one word is sufficient, right? It moves or it pulls the chromosome to the 2 opposite poles. Why? Because nucleus, it has to form 2 nuclei, right? So once the nuclear membrane degenerates, it forms again. Form কৰিবই লাগিব, এটা নিউক্লিয়াছ বা দুটা নিউক্লিয়াছ হ'ব লাগিলে নিউক্লিয়াৰ মেমব্ৰেনখন আকৌ ফৰ্ম কৰিব লাগিব. So whenever we talk about prophase, metaphase, anaphase, and telophase, one line that we remember, maybe about prophase, the first line or the most important line, nuclear membrane degenerates, right? Metaphase of কি মনত আছে? এটা লাইন, বাকিবোৰ ইভেন্টস বাদ দিয়া, just one line. All the chromosome gets aligned or arranged in the equatorial plan, right? বা মিডলত আহি যায়. Next is anaphase. Anaphase of কি হয়? Poles, right? টানি আনে. আৰু telophase কি মনত আছে? Opposite of prophase. Just এক লাইন, right? Telophase, prophase যিমান যি বস্তু হয় তাৰে উল্টাখিনি telophase হয়. তাত নিউক্লিয়াছটো ভাঙি গৈছে, ইয়াত নিউক্লিয়াছটো ফৰ্ম কৰিব. Right? So ইয়াতে কি হয়? এই দুটা বস্তু নহয়, অকমান different from the normal type of cell division. So this is one method because of which DNA content gets increased. Next is we are talking about a specific type of a nucleus that is called a restitution nucleus. Right? Whenever we are talking about nucleus, either we remember a prokaryotic nucleus or a eukaryotic nucleus. Nucleus কিমান type বুলি ক'লে আমাৰ দুটাই মনত পৰে. Right? তাৰ উপৰিও So we have a word restitution. Restitution nucleus ki eta specific condition dekhilehe he nucleus to kami restitution nucleus kom. And what is the condition we are talking about? Soson? Tate jeneke ami unique point to dhoribo parilu no? Yato to okoman kiba ta different hoise. And what is the unique point we are talking about? এই লাইনটো চোৱাচোন, বুজি পাইছা নাই? Is this line understood? If you're talking about the normal type of mitosis and meiosis, tat ki hobo? Yate ki likhise soa. It involves normal mitosis up till anaphase. After anaphase, what is the phase that we'll be talking about? Telophase. What happens in telophase? If suppose we are talking about this as a cell, yate nucleus form koribo, yate nucleus form koribo, right? Nucleus form korile ekhon ki hoy? Nuclear membrane form kore, so this is telophase, normal condition. Yate ki hobo? Right? So duitar genetic material will be covered by a single nuclear membrane. Tat ki hobo? Under respective nuclear membrane form koribo. Is the point understood? Right? As it is, as I said, iman besi important no hoy. Iman questions yar pra no hoy. Just to understand, but just to know, je DNA content ki hor karne badhise, maybe because of such reasons. Right? So endomitosis or karne badhibo pare, a restitution, kisuman plants or ki hor restitution nucleus form kore, a restitution nucleus form kore karne, tapeta mo DNA content badhi jay. Right? So yate ki hoy, this is a condition, or tat ki hoy, spindle fiber form no kore. Next likha number 3. Likha polyteny, Polytene. The third reason might be because of polyteny. Okay, polyteny, hunisa. Ako, when we are talking about what, which chapter? Cell, the unit of life. Last part of the chapter talks about ki chromosomes, yes or no? Nucleus or pisat ki porisa tamanage chromosomes. Chromosomes or special type of chromosomes porisan nai. Salivary gland chromosome, Lambrach chromosome, nai porha no? Ne porisa. Special type of chromosomes to morphology you have to study. Apart from that, 2 special type of chromosomes you need to study. That is called as Lambrach aru salivary gland. Jedi hetu porha nai, then obviously etu gom poa nai. Doesn't matter, we'll revise it later. Eta likhasun. If DNA replication is not followed by splitting of chromosome, the condition or the new chromosome that is formed is called as a polyteny chromosome. The word polyteny means many armed chromosome. Arms keta ase? Bahut gita. Whenever we talk about a chromosome, how does a chromosome look like? Something like this, right? So heard about the, obviously you have studied something called as duplication or replication of DNA. Code porisa, cell cycle, cell division, right? Cell cycle, cell division out. What you have studied, that whenever a cell wants to, is ready to divide or is preparing itself to divide, we talk something about, we talk about certain events that take place in a cell, and that is called a cell cycle, right? Cell cycle has 2 phases. Ki asile? Why are we talking about cell cycle? Because we have to talk about this one. DNA replication. DNA replication, okoman more koribo lagile, we have to talk about cell cycle and cell division, right? Cell cycle ki hoy? Events that takes place in a cell. Which cell? The one that is going to divide. First point. Second, whenever cell is, a cell wants to divide, what are the events that occurs in a cell? Or in other way around, cell cycle is divided into how many phases? Duta phase? Enukara diagram monda sile? right? আৰু এইটো এইটো ফেজক কি বুলি ক'ম? Prophase, very good. Metaphase, anaphase, telophase. So this is called as M phase, and this is called as I phase, interphase, right? Aru hetuk division phase buli koy, right? Interphase ot ki porisla certain events that occurs here, right? G1 tinta phase ase, G1, S aru G2, and you have studied together with them. You have talked about something called as G0 phase also, right? Okay, let's not go to that. G1 phase, S aru G2. What was the most important event that occurred in the S phase? Replication, or maybe you have studied the word called as duplication. DNA duplication. So what's the important question এইটো phase পৰা হেতুৱে আহে যে DNA duplication কোনটো phase ত হয়? DNA duplication সদায় S ত হয়. DNA duplication কোৱা, DNA replication কোৱা. As of now, we'll be talking about the same thing, right? Now the question is, DNA duplication মানে কি? Genetic material increase কৰিব, right? Genetic material increase কৰিব, তাৰ DNA content increase কৰিব. So DNA খিনি ক'ত থাকিব? Chromosomes ত থাকিব. So whenever we are talking about DNA duplication, we are talking about nothing but this one. Yes or no? এইটো the normal chromosome. ইয়াতে arms, আৰু ইয়াতে কি হ'ল? Arms বাঢ়ি গ'ল নে নাই? So polygyny, many armed, right? Normally এতিয়ালৈকে many armed হোৱা নাই. আমি এইটো diagramকো কি বুলি কওঁ? Chromosome বুলি কওঁ. এইটো diagramকো কি বুলি কওঁ? Chromosome বুলিয়ে কওঁ, right? This is a single chromosome. এইটো কি chromosome? দুটা chromosome নে single? This is also a single chromosome. But what is the difference? ইয়াতে duplication হোৱা নাই, তাত duplication হৈছে. ইয়াতে duplication ক'ত হৈছে? S phase ত হৈ গৈছে, right? Now coming to this part. So polygyny কি হয়? The DNA will keep on duplicating, but the chromosomes will not split. So what you will see is, okay, chromosome বুলি ক'লে, this center is called as কি? Centromere, right? It is that point of attachment of the two. Etu ki hoy? Etu ki hoy? Chromatid, right? So ketaman word ase no? Janu? Talking about chromatin. Chromatin kwa ba chromatin? What to do now? Okay. Okay. Talking about chromatin network. Thread-like structures, right? Karan ki hoy? We know inside the nucleus you see genetic material. Genetic material in the form of what? Chromosomes. Kintu prothom dinai chromosome formot thakibo? Ne nathake. Obviously nathake. Genetic material or chromosome buli ketia koba? When they will have a proper morphological shape. Prior to formation of this shape, genetic material kenuko asle? Thread-like. Right? Jetia eta nucleus ase, nucleus or bitor eneke thread-like ase, thread-like genetic material okami ki buli kom? Chromatin. Chromatin bilage ki form koribo? Chromosomes form koribo by a process of what? Condensation. Right? Condensed hoy jabo, horu hoy jabo, horu hoy gole tar morphology tum bhalke dekha baba. So this thread will be forming this one, or maybe, to be precise, this one, right? This part is called as ki, sorry, centromere, aru eigita ki buli koy? Chromatid. So what is a chromatid? The arms of a chromosome is called as chromatid, right? So yate so polygyny condition at ki hoy je DNA replication hoy thake ba duplication hoy thake kintu separate no hoy. So you'll see a centromere with many arms. Is the concept clear? So polygyny chromosome eta dekhat kenekua, something like this. Normal chromosome eta dekhat kenekua. Like this, right? So DNA duplication hoy thake kintu ebilak ki hoy split hoy na jay. There is split hoy na jay etate bahut arms thakibo, and that type of a chromosome would be called as polygyny. And the one thing that we know is whenever the arms are more, obviously DNA would be more. So tapetal cells are maybe because of this reason the DNA content is more, right? So ekhini asile okkoman extra type. Kinetochore. What is the question? No. Kinetochore ki hoy? You have the centromere, right? You have to disc. Right. So kinetochore boli kole disc-like structure that surrounds the centromere. First point. Second point, it is that area where the spindle fibers will get attached. Right? Spindle fibers bilak ami normally keneke draw koru. Ami suppose we are talking about this as a centrioles. It is a spindle fiber hoy. Yate ami chromosome to aki disu no. I think you remember from cell cycle cell division. Right? Ako yate eta chromosome thakibo, yate thakibo. Right? So spindle fibers actually coat lage. In general, we say that chromosomes are lagi jay, but centromere, actually centromere na lage. Centromere covering ase, that is called as kinetochore, that is chromosome to likhe. So question from cell cycle cell division A2 ahe, the adult chromosome kinetochore keta, duta. Right? So you have one disc, one covering this side, and the other covering this side. Right? Whole, no? Okay. So what we are studying is we are talking about the formation of, or we are interested in studying about the male reproductive part of a flower. Up till now what we have studied is microspores. Etyalake form kora nai ne korile nai kora okol pollen sac to form korise. So the next topic that we will be studying is we will be talking about how is the microspore form. The one thing that we already know is who will form the microspores? The sporogenous tissues will form the microspores, right? Whenever we use the word sporogenous tissues, it a question ahibo regarding ploidy, right? Ploidy jano no, haploid diploid jano. Question keneke ahe, what is the ploidy of sporogenous tissues? Question ahibo, what is the ploidy of microspores? Ploidy buli kole, I think you have heard two words. One is called as sporophyte, the other is called as gametophyte. Plant kingdom or kunisila ne words kita? Sporophyte, kisuman plants ok, ki buli kom? Sporophyte buli kom. Kisuman plant body kami ki buli kom? Gametophyte buli kom. On the basis of what? Why am I talking about this sporophyte and gametophyte? Kan ploidy bostu to concept to tumalke 11 or porhisa. On the basis of either it's a haploid condition or it's a diploid condition. So each and every cell is a haploid. The entire plant body would be called as a gametophyte. Protect gute plant dalor protect to cell except few. Zedi diploid hoy. Tela gute plant dalor kami ki boli kom sporophyte, right? So sporophyte bilakot kenuka dhoroner cells take twice n cells take diploid. Sporogenous tissues or a question ahibo sporogenous tissues or ploidy ki. Sporogenous tissue is a part of which plant? Angiosperms. Angiosperms is a gametophyte and a sporophyte. It is a sporophyte. So if it is a sporophyte, cells bilakor ploidy ki twice n, তেতিয়াহলে sporogenous tissues or ploidy ki twice n. Is the point clear? So tenuka dhoroner questions aibo. Again the question could be, what is the ploidy of the tapetal cells? We have talked about a word called as polyploid, right? So polyploid condition, thik ase tapetum to jodi nokoi pelai, if I talk about epidermis, epidermal cells or ploidy ki, epidermal cells first car epidermis, angiosperms or epidermis, angiosperm or plant or body ki, sporophyte, to ploidy ki hobo, diploid hobo, right? Okay. So etia ami ki porham, we'll be talking about the next step. X again. Yes? Now, in the entire plant kingdom, no? Plant kingdom or pasta group of plants as no? Algae, algae, bryophytes, pteridophytes, gymnosperms and just leave aside algae being the most primitive plant. If you consider from bryophytes onwards, bryophyte okol gametophyte. Baki pteridophytes onwards, so boke, ami sporophyte boli kom. So pteridophyte or plant body, sporophyte, gymnosperms, sporophyte, angiosperms also sporophyte. Right? Okol bryophytes or plant body to ki hoy? Gametophyte, right? Okay. So the next topic is microsporogenesis. Microsporogenesis would be talking about ki, the formation of microspores. Right? Microsporogenesis key, it is a process through which the microspores are formed. Who forms the microspores? The sporogenous tissues, right? So we'll write the definition first. So the process of formation of microspores is called as microsporogenesis. Who will be forming the microspores? We have used a word called as sporogenous tissues. Instead of sporogenous tissues, we can also use a word called as, yes or no? It could be also called as pollen kune form koribo, pollen mother cell form koribo, pollen mother cell ami ki buli ko, microspore mother cell ko paru, and microspore mother cell kune form koribo, sporogenous cells bilake form koribo, so it's the same thing. Now the point is you have to choose between these two words. Read the sentence, choose between mitosis and meiosis. Sentence to porha and see whether you have understood or not. What we are talking about is microspore formation. Kune form koribo, microspore mother cell form koribo. Microspore mother cell jodi microspore form kore, so obviously it has to undergo cell division, right? So what is the type of cell division that they will undergo? Mitosis, meiosis. Meiosis. From reproductive point of view, obviously it is meiosis itself, right? So they will be forming through meiosis, okay? So we'll have microspore mother cell, right? Undergo ki meiosis. What they will be forming? Microspore, which is also called as pollen grain. Now coming to this, we want the ploidy of the two cells. Any cell that undergo meiosis is considered as a haploid ne diploid cell. Diploid cell, to MMC ploidy ki hobo? Twice n. After meiosis, they will be forming a cell, and that cell would be haploid ne diploid. Question to buzi paisa? MMC ploidy ki? Twice n. This cell, any cell that undergoes meiosis would be called as ki? Meiocyte. ঠিক আছে এতিয়া ইয়াতে meiocyte কোনটো microspore mother cell, since it undergoes meiosis it is called as a meiocyte. ঠিক আছে বাৰু এটো যদি কনফিউজন হৈছে এটো বাদ দিয়া microspore mother cell, pollen mother cell we understood. ঠিক আছে এই সেলটোৱে will be undergoing meiosis and would be forming কি microspores. What is the ploidy of microspores? What is the ploidy of a spore? Spores are always haploid. The spores are always diploid. Spores are always haploid, right? Maximum case of spores ki hoy haploid hoy. Ar eneo meiosis mane ki? Meiosis is also called as dash division, reduction division. What gets reduced? Mitosis is called as ki equational division, right? Meiosis is called as reduction division. Equational ekei thake ki ekei thake? Number of chromosomes ekei thake. Mother cell and the daughter cell, the number of chromosomes remains the same. If you are talking about meiosis, the number of chromosome gets reduced. So from twice n reduce ki hobo? Na2 hobo. Is the point clear? So whenever we are talking about pollen grain, whenever we are talking about the word spore, except a few spores, আমি তোমালোকে এটো ফানজাইট পঢ়িছিলা, except a few spores, whenever we use the word spore, spore is a product of meiosis formed by spore mother cell. So obviously spore ploidy ki n, gametes or ploidy ki n, pollen grain or ploidy ki n, egg obviously n. Gametes or jodi ploidy n hoy, pollen grain ki hobo? Tar bhitorte to male gamete to thakibo, right? So I think it's understood now. So pollen grain is haploid. Pollen mother cell is diploid. Okay. Next, another point about meiosis. If a cell undergoes meiosis, what does it produce? Daughter cells. If a cell undergoes mitosis, then also the cell would be producing daughter cells. The point is, how many daughter cells in mitosis? How many daughter cells in meiosis? Exactly. Mitosis যদি হয় এটা সেলে কেটা সেল ফর্ম করব? দুটা। যদি meiosis হয় এটা সেলে কি ফর্ম করব? চাৰিটা। So this cell would be forming কি? 4 microspores, right? And so later we'll be using a word called as অলপ পিছত আমি এখানে ডিটেইলছত লিখিমেই। লিখাৰ আগতে অকমান জাস্ট বুজি লৈছো। Right? So MMC asile, MMC meiosis undergo korile, ki form korile pollen grain. At one division or per division, keita pollen grain ekelege form koribo, saita. Jehetu saita form kore, we'll be using a word called as ki pollen tetran. So eta division te saita pollen grain form kore. Clear? Okay. Next, Swan. What we have said, if suppose this is the tapetum, we have already drawn the diagram, no? If this is the diagram, if this is the tapetum, inner to tapetum ki asile? Cells, right? A cells bilakok ki buli kom? Sporogenous cells, or we have new names now. Sporogenous cells bilakok ki buli kobo paro? Pollen mother cell ba ki spore mother cell olof agote likhisilu ta mane eketai bostu, right? So what we'll discuss is yar bitor jiman bor tissues ase no prottektuke ami ki buli kobo paro? Pollen mother cell. So now we understand that each and every cell has the capacity to form pollen grains, right? অকল এটা সেলে pollen grain form কৰিব নে গুটেইকিটা সেলৰ capacity আছে গুটেইকিটা সেলৰ capacity আছে but we will see that out of all the cells that are present some of the cells will die যিমান cells আছে capacity কাৰ আছে গুটেইকিটাৰ আছে কিন্তু গুটেইকিটাই form কৰিব নকৰে কিছুমান cells will die so later what you will see if so maybe is the point clear আমি এতিয়ালৈকে process to লিখা নাই process to আমি next class ত লিখিম right so what we will study that we have talked about the tapetal layer inner to tapetal layer you see a collection of cells that is called as pollen mother cell right all the cells of the sporogenous tissue বা all the cells could behave as mother cells কিন্তু গুটেইকিটাই divide কৰে নেকি নকৰে কিছুমান cells will degenerate and they would be providing nourishment to the dividing mother cells right so this is a portion that we will do in the next class okay and together with that we'll discuss certain questions also ha ajiloke jikhini porhilo ketaman question korile clear hoy jabo thank you